So that's a bit on CIRM. I would like now, I think we're right on schedule, Bernie. It's my pleasure to introduce, every organization has to have a person at the helm who is a person who commands great respect in the area. We are very fortunate to have as our president a world-class scientist in the stem cell space, somebody known internationally who has helped, among other things, forge collaborations with 11 countries who are putting up their best and brightest to match those that we have in California towards progressing stem cell research. He oversees our staff of roughly 40 who are doing, I must say, a terrific job. If you were to look at the programs that we have in place, the programs that our science staff administers, and that are moving along in the utmost professional manner, Alan is, of course, the head of that effort and is the face of CIRM throughout much of the world. It's my pleasure to introduce, to give the keynote speech here, my colleague, and before I say that, Bernie, thank you so much for all you do. This is a wonderful event, year in, year out, bringing together all the many stakeholders, and I salute you for what I know is an incredible amount of work. So without further ado, my colleague, Alan Trounson.
I didn't recognize myself. Thank you, John. And welcome everybody to the session. It's going to be an interesting 3 days, and I want to thank Bernie for responding to a few cues that I gave him, and then for inviting me to open the meeting. But I told him, I thought, how would you distinguish the meeting at the moment? And I said, there's such a momentum going on in translation. You ought to create a focus for that, because the hardcore science is done very, very well in the International Stem Cell Research Society. But this is an opportunity to come here and be doused in the translation that's actually happening in California. And so he responded beautifully, I think. And so I think you'll enjoy this meeting, because it's fun, because things are happening. The research is rolling over to the applications, and you'll see the traumas as they go through to clinical trials, but also the potential of the whole program. And I want to thank John Thomas too publicly for stepping into the shoes of Bob Klein. Big shoes, all right. Bob was a great friend and a genius and hard work. And John is a listener and a negotiator, and it's been a really easy transition for most of us. And I thank him for being thoughtful and helpful in getting through that transition. So I think we've got a very functional outfit. What we've got to do is get on and deliver. So this, let me just see if I can make this work. Okay. So just a brief word about where we are. We have over 450 awards with over 1.5 billion committed. So if you look at it, we're heading towards the last third of our funding, and that's fairly significant. And so at the moment we're actually revising our strategic plan as we're required to under our schedule of the original strategic plan. And so we will create another degree of focus, and John mentioned some of those. Clearly business and connecting with the business world is going to be important, but we've got several new really major initiatives that are underway, and the stem cell leadership in California is aware of that now. We have over 900 published papers, and I think it's close to 950 now. And we only started funding at the end of 2006, so that's phenomenal. But what's more important, I think, is that 24% of them are in the high impact factor journals. So California is having an overwhelming influence on stem cell research in the world, and I think it's driven pretty much because of the science is really up and pacing very, very nicely. So, I mean, I'm not going to talk about the basic science, but that's just phenomenal what's happening in that space. I'm going to talk more about the translational work, but you know, altogether, this is really quite a phenomenon. And when I took over this job 4 years ago, I thought, well, maybe we could run a revolution in medicine, and now I'm thinking we're likely to deliver on a revolution in medicine, and I hope you will agree at the end of these 3 days. The new lab's dedicated to stem cell research, hundreds of young people trained in future jobs right across California, out to schools. We now have a program where I initiated taking high school students in and giving them a chance to work over summertime with the stem cell labs, but also at the same time music or ballet or physics or chemistry or something different, to bring in that creativity that young people have and see if we can initiate that creative surge that happens in that space through to the Bridges program, which draws on those Californian institutes where a lot of under-resourced students are, who don't normally get the opportunity to move into the top-line laboratories. And I think both those programs have been outstanding successes. There are more than 24,000 jobs been created and $2 million in tax revenue, 800 million plus in leverage revenues, and funding relationships now with 12 countries. So these are the bars for our funding, and I hope you can read that all right. I'm going to look over the shoulder to see if you can see it. But we've been moving those bars through those programs. So there's very small amounts spent, very small spent on administration. The majority in the early days was really for training, and then for establishing the institutes, and then the lighter bars come on and basic research, and then the very light bars in translation. So you can see that in these bars that we're opening up the whole program. Basic research is still being strongly funded, but we're moving more into translation. So there are $360 million plus now in 44 research projects across 26 diseases in this clinical area. And it includes embryonic stem cells as well as adult stem cells. And you'll note that the embryonic stem cells does not overwhelm the adult because there's some real opportunities, particularly nearby, to get work, go to the clinic, and we'll describe a number of those projects during these 3 days. Cancer stem cells, a horrifically difficult cell to control, to identify. We're after that cell. We want to take out the metastasizing cancer stem cell, and we're putting a lot of effort behind that. Our Canadian colleagues are working closely with us to do that. IPS cells, the induced pluripotential stem cells, are growing and will continue to grow, in my view, will continue to be more and more influential in the area. And also, I think with the new developments in the way you can actually transit cells in the body from one cell type to another, I believe that the future platform for stem cells will probably shift to more of the inducing the endogenous stem cells to change their nature from one cell type to another under the influence of transcription factors and also cleverly designed strategies and targeting using viruses to move cells into regenerative capacity within the tissue. And this phenomenal new work is really now starting to build a whole new platform for regenerative medicine, and it's wonderful. So I see partnerships in translation of preclinical research as being really critical There's the academic, which is required for research infrastructure, for the research capacity, innovation, intellectual property, and the depth to solve issues as they arise. So this partnership, when together with industry and their manufacturing capacity, their standards, their focus, and criticism often in academics is they don't focus, they don't hit those milestones and go, no-go decisions. Well, they're kind of learning that sort of stuff pretty quickly, so you can train these irascible clinicians and academics to do that. But it also helps to have partners in the industry to do that. And I think relevance and really, what's a competition doing is really important. It's no use developing a treatment that's got a better competitor, or at least a good competitor, because it won't actually succeed in the long term. Utilising IP, a lot of IP is left by academics because they don't utilise it. And of course, clinical trials is really the bread and butter for the companies. And the clinical medicine, without the clinicians and reverse translation back through to the academics, I mean, what is meaningful? What do you need to deliver to the patient? That's what's really critical. You need that message come back. There's no use developing a fantastic treatment. It's got no applicability because the clinicians won't use it or can't use it. So they've got to be involved. Patients have got to be involved, the advocates, the patients themselves, clinical infrastructure. Counselling. In this area, you need counselling. I'm very strong about that. I saw the need for counselling when I was in vitro fertilisation. I see it here. You've got to help the patients understand what's really relevant to them and what can they access in this space. And monitoring the patients in the long term. When you give a patient a drug, it decays away. When you give a patient cells, they could develop and continue. You need to have a long-term monitoring to make sure you don't get a surprise 5 years, 10 years down the track. And without that infrastructure, that medical infrastructure, you won't know. So this is the way we've kind of built the programs. I have a view now that in many of these projects, the sun never sets because we have these collaborative funders, and they're working. The sun is up around the world 24 hours a day. You see the Californians are linked to the Canadians, and you can link to the Australians, link to the Germans. These linkages are there now, and so the project is actually going 24 hours a day, and they talk to each other. And so how the results go? Oh, it didn't work. Oh, we'll do it. No, we won't. We'll do it. And it's driving that. It's actually driving that system harder and faster than ever I thought was really possible. And if you look at in the red are the collaborative funding arrangements that we've got, the agreements set up there. These are the scientists in these countries can work with the Californians. They can work together with others as well. We can have 3-way or 4-way agreement. But here they are in red, and we've got them. This is a network of the best scientists in the world now. So you can go work with those scientists. You apply to CIRM, and we'll pay for the in-California part, and I'll pay for the out-of-California part. We've started to get the states involved. They're in the green, and the foundations, the JDRF, and Maryland is already put in for 2 projects. I have just signed an agreement with the National Institutes of Health, so we're going to get them in the paddock too. That ought to be good. That'll be interesting. I mean, they've got some phenomenal capacity in their clinical areas. Mahendra Rose just moved there. We've got all the ingredients to turn this network into a powerhouse, a huge powerhouse in regenerative medicine. And I think it's going to work. So going to the clinic, we see we're in the early stages because we're sort of backing their new initiatives. So you have this sort of proof of concept there in the early stages, the feasibility. And then you have the development of candidates. When the scientists say, this is a candidate, this is the cell, this is a molecule we want to do. Then we've got to move it up to get a registration with the FDA, and then move it into clinical trials. And we've been working hand in hand with the FDA ourselves through an organisation that Ilona Baum helped set us up with the FDA, and it's been a fantastic to and fro with the FDA. We're learning from them, they're learning from us, and now the Alliance for Regenerative Medicine has clicked in as well. This is a very much a hand-in-hand process, and we're learning, and we're actually, we're being stimulated to think, well, what do we need to sort of solve that problem, or is it a problem to solve, or, and the help of the regulators instead of seeing them as opponents is terrific, and it's actually working really, really well. So hopefully we'll have an effective system and a pathway set up. We're working on a range of these frontline diseases, and the ones I've circled are embryonic stem cells. They're circled in blue. The one in green is the first one that's moving out from induced pluripotential stem cells for a horrific disease called epidermolysis bullosa. But you can see that we're looking at cancers with genetic problems, neurodegenerative disorders, and this is the kind of front line. It's stressing us, it's stressing the scientists, it's stressing the teams as we move up this pipeline, because it's hard work going up that pipeline, solving the issues, meeting the pharmacology, the toxicity, and we are hitting rough spots, but we're making our way through it, and most of the teams are going really, really well. And those that are not, we're trying to revamp, trying to refocus, and trying to get them to lift in a different way. So in the gene therapy area, I actually think this is kind of low-hanging fruit. This is going to work really well. Fanconi anemia, that's while it's in that, it's in that early stage. That's, that disease with the scientists at the SOC using autologous genetically corrected iPS cells. I think it's going to work, but also the ones that are, further downstream, sickle cell disease and epidermolysis bullosa, they're heading towards the clinic, those. And what they do is they're taking the cells, in the case of hematopoietic stem cells for sickle cell disease, they're correcting that. And I think you'll hear Don Cohn talk about that during this meeting. And if you want to see where the sort of hard front edge of this work is, you should listen to what they're doing. I believe that they can move very quickly from sickle cell disease to thalassemia. And thalassemia is a horrific problem. And if there's a cure for both those diseases out of the same piece of research, it should be wonderful. And Duchenne's muscular dystrophy for the researchers at Stanford, that's in the feasibility studies. It's a terrible muscle disease, and the need to treat that, and the focus there is autologous skeletal muscle precursor cells that are derived from the human iPS cells, are genetically modified again to correct that gene. This work, I think, is going to happen. It's going to work. In the case of that epidermolysis bullosa, you can see that horrific scar on that young person. What happens is that there's a defect in collagen, so the skin, the epidermis, won't attach to the underlying dermis. You wipe these kids and you wipe off their skins, and there's you go listen to, I think this is now on their website, a terrific talk by John Wagner. On this disease. This is a disease that parents, just really, really, really need some help in. It is a case where some mothers have cried at night hoping that their child will die rather than continue in the terrible pain of having what's equivalent to burns the whole entire time on their body. It's just a horrific genetic problem. And this, I think, will be overcome with this strategy. Well, I think the bone, cartilage, and muscle area, where we're working with about $20 million. This is in the very early feasibility studies, and there's osteoporosis, a terrible problem for bone-breaking disease. Looking at the way of being able to correct that to the scientists at Cedars-Sinai, look at the feasibility of that. But the ones in osteoarthritis, and they're moving fairly quickly in its scripts, and in those two diseases, the focus of those studies looks, really looks like it's going to work for them, I think. So I expect one or several of these studies to go off towards the clinic shortly. And the spinal fusion work at UCLA, again, it's terrific work. Cancer, we're investing over 100 million in this, and we're after this horrific cancer stem cell that metastasizes and kills people after they've had all their chemoradiant therapy. This cell stays in a niche. It's a quiescent cell, and it's really hard to get. So we're focusing on strategies to try and take that nasty cell out. So there's a whole range of diseases, cancers that are in this sector, with hematological malignancies being high in the order, and glioblastoma, which is the brain tumours that kill so rapidly and efficiently that just a terrible disease. So these are the diseases that we're focusing on, and you can see that these are more advanced. So we're in the more advanced stages, heading towards the clinic, and we have very good relationships with Germany and in Canada and the UK in these studies. We're bonded together with the scientists in those countries, attacking these problems. So it's through small molecules, monoclonal antibodies, figuring out the nature of this cell, trying to get into it, target it, take it out. We'd prefer to take that cell out than the rest of the tumour, because the rest of the tumour probably can't seed the cancer again. It's these cancer stem cells. Whether they make up 30 or 40% or just 2% of the tumour, we've got to get rid of them. And leaving anything behind is a legacy for death in the future. So this is an important strategy. One of the front-running projects is to take neural stem cells, load them up with a gene that can convert a drug given to the patient to a very, very toxic component. So if you look at the way they load up these neural stem cells, and with a molecule that can convert this one to this very toxic SN38, a very, very cytotoxic drug. And these neural stem cells will actually home to the tumour. You inject them to the patient, they'll home to the tumour, they'll gather around and inside the tumour. And then when they convert this drug given to the patient to the highly toxic drug, then what happens is it kills all of the cells in the nearby vicinity, including the cells that you've given to the patient. So, I go back one. So this is Karen Albert Bitties' study, where when you give up in the top line, this is the animals with the tumour, and this is after treatment with those cells that are loaded, that have been transfected with this gene that can convert CPT to the more toxic substance. And if you just give the cells only, or if you give the drug only, you can see either the disease will remain in a stable way. Let me just see. It'll remain in a stable way, or the animal will die. And if you give the cells together with the pre-toxic substance, it wipes out those tumours incredibly effectively in the brains of these animals. And so she's got some work going on with a less toxic substance already in the clinic, and we're funding her to take this really, really powerful substance there, so we can actually get into those tumours in the brain, these inoperable gliomas, and take them out using this methodology. And any progress in that area would be fantastic. The eye diseases are critical. As we get older, we all lose that central vision in terms of macular degeneration. It's affecting 24 million people. So we have a lot of projects in this area. So the early projects on limbal cell deficiency is a feasibility study from the group at UCLA. But age-related macular degeneration is being attacked with 3 projects. One of them is moving very quickly to registration, or hopefully submission for an IND registration. And there's also a very interesting study on retinitis pigmosa, and I talked to the scientists at UC Irvine And I talked with them just a month or so ago, and that project is really ready for going to the preclinical studies now. And it looks like another effective strategy. So the eye is a place which is going to be effective. So this is the studies by my Mark Humanen's group, where they're putting these cells, embryonic stem cell derived retinal cells, onto a parallel membrane. Here the cells are on that membrane, and they'll stay on that membrane. They won't migrate off. So it's like a little stamp. And with those cells, they're on monolayer. You can see them, they're on the monolayer. If that can be just inserted into the correct position, then you get a very effective bonding here, this monolayer. This is the new retinal monolayer there, and it's perfectly connected to the cells in the photoreceptor cells and into the underlying membrane. There's a perfect adhesion and a terrific connection. That's exactly what you need. And that's in the central spot where you've lost that central vision. So this looks like it's going to work in my mind. It's a very smart technology, and they're getting close to going to the clinic, or at least applying for an IND. The neurological disorders, we have almost 100 million in that area, and goes from Lou Gehrig's disease, ALS, to Alzheimer's, Parkinson's disease. And there's a big commitment in these projects. There are three projects focused on Parkinson's disease. Autism, Huntington's disease, there are 2 projects in Huntington's disease. And spinal injury, there are 2 projects in that area. So we're working across that space. There's also studies on epilepsy, caravan disease, and spinal muscular atrophy, as well as stroke. So you can see this is a broad spectrum for us to work in, and the studies are challenging because they're still in an early state. Correcting neurological and degenerative diseases is very complicated, and it's requiring very astute scientists and very astute science. And you can see the number of Parkinson's disease projects, all different kinds of approaches to there. The Salk and Germany are working together on the feasibility study with small molecules. Burnham is looking right across the space, looking at adult stem cells, embryonic stem cells, or iPS cells, which is the best cell to use in this, and they're working on non-human primates. There's autologous HSP-derived dopaminergic neurons being studied at the Buck Institute, and so these are embryonic stem cell derived or iPS cell derived cells. With Huntington's disease, an approach trying to block the effects of those huntingtin protein, and also Alzheimer's disease. So there's all this work going, and it's moving in that developmental phase, trying to actually get it ready for the preclinical studies. Neurological disorders, caravan disease is for young people. Autism spectrum disorders, again, a problem for young people, babies and very young children. We're looking at refractory epilepsy and spinal muscular atrophy, and ALS is up in that area where we're trying to push that work led by the UC San Diego group to the clinic, moving that ALS towards the clinic with an approach growing embryonic stem cells into astrocytes and using those astrocytes in the spinal column to try and correct that really, really difficult disease of ALS. So these are good studies. Now, neurological injuries, there's spinal cord injury using embryonic stem cell derived motor and autonomic precursor neurons. It's in the feasibility studies with the UCLA. There's a spinal injury we're working together with the Geron Corporation now, and that's in clinical trials and phase 1 studies. The 4th patient was done a little while ago at Stanford University. So this is moving forward. We've got great hopes that this work will progress. There doesn't seem to have been a problem that we've heard about at all with the patients that have been treated. So these are safety studies. Of course, they've got to move on beyond that. We're also interested in traumatic brain injury and stroke. Stroke is the other project which we have high hopes from Stanford, using embryonic stem cell-derived cells to have a positive effect on those patients who've had a severe stroke. The HIV area is an area where I think we are going to have a cure, I think almost certainly. And it'll come from probably one of those two front-running projects from UCLA or the City of Hope. But there's also a very interesting developmental study that's also being run out of the City of Hope using autologous hematopoietic stem cells that are genetically modified as well. So if you work in this area, and I'm only going to take one example here, and it doesn't mean to say that the other studies is or isn't going to work well. The other studies using short hairpin RNA interference, and I think that's a really smart way of trying to take out a gene. But these zinc finger nuclease technology looks to be very effective. It's being used against T cells at the moment to knock out a co-receptor, a gene as a co-receptor for the virus CCR5, the gene CCR5. And you can design these zinc finger nucleases, and it's done by the Sangamo company, who've got very beautiful design technology for this, and very, very accurate, very effective, and it looks like it converts a lot of cells when you treat them. So the target is the CCR5 gene. It's a co-receptor for the HIV virus. And it's known that patients who've got the CCR5 mutation are totally resistant to HIV infection. That appears in patients in Scandinavia, and they do not get the infection. And a very smart clinician from Berlin went out to find a mutated CCR5 donor and gave it to a patient who was really sick with an AIDS-related cancer. That patient has totally recovered from the infection. And so this Berlin patient is a proof of concept. One patient, but a proof of concept. So the studies that they've done in the animals are confirming that you can actually remove the virus cannot live in the state where the cells have got this mutation. The co-receptor doesn't allow the virus to infect the animals. So I expect this to move into the clinic very quickly, and hopefully it'll be a cure for HIV. And I have a view that we shouldn't stop there. We should think about that as an immunisation strategy and take it to the world. And why don't we eradicate in one generation this horrific disease? Why don't we do that? If we did that alone, it would have been, a massive milestone and worth every single cent of the $3 billion. Diabetes, the study on diabetes, there's a wound healing study at the UC Davis and combined with the scientists in Germany. And the ViroCyte company has got a way of developing embryonic stem cells into insulin-producing islet-like cells. And if you put them into what looks like a credit card, a way of encapsulating these cells, the animal, the patient's own immune system cannot attack them. And so they'll produce insulin for the period of time those cells are there. In animals, it's correcting diabetes. It looks like a terrific project. There are other applications, liver failure, advanced ischemic cardiopathy, beautiful work being done at Cedars-Sinai Medical Center, and that's an autologous cardiac-derived cell treatment. I think that's going to work. It looks very effective. It should get an IND, I think, in the near future. And then there's an approach using recombinant Wnt in a sustained release formulation to stimulate endogenous stem cells to repair tissue from Stanford. A very, very interesting study which is looking broadly across all stem cells. So this is that's the whole deal. We're looking ahead. There's 19 researchers assembling therapy in development teams that we expect to come forward in 2012, more teams working towards developmental candidates, more teams working towards IND and phase 1-2 studies. We don't think we'll go into phase 3. We don't think we've got the funds to do. That's why we want the companies to link with us and provide that opportunity to go the distance. And lastly, I want to say that this is really challenging, tough work. You take your discovery, you've got to get through the regulatory pathway, you've got to address all the regulators' concerns, and working in hand with the regulators, develop a product which may need commercialisation. Very probably aspects of it will require commercialisation. Get it into the clinical setting and then get it to the patients in clinical trials, and then hopefully, if it's effective, into a clinical format. I believe it's going to also take us to set up stem cell clinics in California, and I have a commitment to look at this, and I want to see stem cell clinics, a network of them set up in California, and I have colleagues now who sort of studying how this could be developed, because we want not only the opportunity for these clinical trials, but these clinical treatments. And if nothing more happens, for some reason we don't get any more funding, there's going to be a strong, firm footprint left in California from CIRM, and everybody will remember it. I hope so. Thank you very much.